All right, I know people are eager for lunch. We've been running a little bit behind, but we have one more presentation from Combined Brain. And, again, on the theme of using the samples that y'all are giving to get insights, especially around biomarkers. And I'll hand things over to Lindsay. She is the Chief Scientific Officer at Combined Brain and the mom, also a rare mom.
Great, thanks, Charlene. All right, thank you, guys. How do I advance this? Green button, perfect, okay. Luckily, when Charlene asked me to speak, I saw I was right before lunch. So this is gonna be quick, there's not gonna be any data. I just wanted to address our new study that we have ongoing that involves a lot of your community members here, and answer some of our basic questions that we have from you and other families that are in this study. So talking about more of an integrated multi-omics approach that we have going on at Combined Brain. I'm the CSO, Lindsay Wieczorek, and I'm also the daughter of a Singapian. So this is our study. It's called Biomarkers. This is the current study. I'm not talking about the other ones that we've done because Jim covered that very nicely. But overall, our goal, again, for this one is to accelerate biomarker discovery, clinical trial readiness through integrated analysis of biological and phenotypic data. This includes 13 different disorders. STX is one of them. And it includes 20 individuals per disorder and a lot of controls. We're going to be talking about different omics data, or looking at different omics data and natural history data. I'm going to go into mostly what that's about. And so the overall goal or the analysis would be to reveal shared or disorder-specific molecular signatures, looking at cellular pathways, looking at circulating proteins, and associating that with genetic variants and clinical features. So these are kind of the big areas of like, what is it and why do we need to do this? So this is just kind of a very basic view of what the molecular foundation of health is and disease. So during typical development, you have DNA, which serves as kind of that blueprint that stores all of your genetic information. From that DNA is produced an RNA, which carries the information out of the nucleus and starts producing those proteins. So we're talking about proteomics analysis. That's looking at the proteins there shown as the machinery that carries out the cellular processes. So when Jim was talking about 10,000 proteins that we measured, those are all those different proteins that are produced from the DNA, from the RNA, and now they're proteins, and they make up your whole person. So that those are the general outcomes, the shape and the cells and the tissues and the traits in your health are all generated from your DNA. So when we have an event where there's a mutation, that's what's all bringing us into this room. My daughter's specific mutation is a 4 base pair deletion, which is shown here, and that leads to a neurodevelopmental disorder. Her mutation is in the SYNGAP protein, your mutations are in the STX protein, so kind of a similar thing where we go from the typical development onto a new route, so into a neurodevelopmental disorder. Where there's a mutation, it's not necessarily always a 4 base pair deletion, but in this case, it resulted in a truncation of the protein. Other disorders or other mutations can kind of do the same thing. So you can see that the route for the neurodevelopmental disorder, now the RNA is looking a little bit different. The protein has a little bit different shape to it, and the function is also going to be a little bit different. You're seeing this in kind of the different tissues and the neurological function that your kids might be going through. So now we have to ask new questions. This isn't a typical development. We have to ask the questions of what happens once you have this mutation. So how has this genetic information changed? This is something you're always going to have the same DNA. Sometimes how it's expressed changes once you have a mutation. So are there changes in expression of the mutated gene, or are there changes in other genes? And how does that message change then when you go to produce that RNA? Is it still produced? Is it degraded? How does that change once you have a little bit of a difference in the way that the DNA is produced? And then proteins, how is the protein machinery affected? Jim and Mike were going through looking at those different proteins and kind of the different levels of expression that might be changed in STXBP1. So how does the levels change? How does the function change? And what mechanisms associated with those proteins change? And then the biggest things that you all deal with every day is how does that person change? How is that person changed with this mutation? What are their symptoms? How do things change over time? So when we think about comparing these things, we have to look at the typical development compared to neurodevelopmental disorder, and we're doing that based off of looking at your blood. So in this collection event, we're having a collection event here, and we're asking you to donate blood. We're going to process that in different ways to be able to look at this multi-omic study that we have going on. So in taking your blood, we're looking for participants that are typically developed, healthy controls, siblings is what we're kind of targeting, and those that have the neurodevelopmental disorder, your STXers. So we're taking the blood and going to be evaluating genomics, transcriptomics, and proteomics for this study. So genomics is just the evaluation of your DNA. What is the mutation? What is the specific mutation associated with the disorder? How does that, for moving into transcriptomics, how does that affect the overall RNA expression that's going on within your disorder? And then, again, the proteomics, which Jim and Mike introduced very lovely. So the proteomics overall, kind of what are those expressions? And it just shows that we need the control participants just as much as we need the affected participants, even more so because the affected participants are a very rare population. You all are aware how rare they are. So we need a lot of control information in order to be able to use that as a benchmark. And one thing that's very vital for this community, donating blood is one thing, but being able to associate that with the symptoms and the outcomes of the disorder is really something that you uniquely can provide. So what we're asking for within the study that we're doing, and for anybody that's interested, is providing that symptomatic information, filling out surveys. I know that surveys is always a hard thing to think about because it does take time, but your community has a dedicated combined brain research coordinator. They've likely been reaching out to you if you're involved in this study, and they're asking you to complete a series of surveys about your child and your experience with your condition. So those surveys include the ClinGen Health and Development Survey, which is really kind of a broad overview of what health looks like, what systems are affected, going more specifically into like specific domains and disorders in the ClinGen level 2 and 3, going into some health history, those types of things. There's another survey called the Vineland. I'm sure you've all probably done that. That looks at more of the functional aspects of what does daily life look like. There's a PELS survey, which is an epilepsy survey, and a diet survey. All of these things really help capture the understanding of what your daily life is like, what your disorder is like, and how the impact of this mutation, the DNA, has really impacted your lives. So this is all associated back with the proteomic data, with the transcriptomic data, with the genomic data to create a holistic view of what is going on within this person's biology. So these are things that we do have remotely available. The symptomatic work is done through surveys that are available internationally, globally, domestically, and wherever you want to be, you can fill out those surveys. We can do blood collections in-home. Within the United States, we do collections here, which is we're here to support you and answer any questions and collect your blood today. And we can also do those at home, and you can attend any of our road shows around the United States as well. So all of the data will be provided back to your patient advocacy group to utilize for research in whatever way is most appropriate for your condition. I'll also mention when you're donating samples today, we just need a little bit of those samples for the study. The rest of it goes back to the biobank and is available for other research studies. So donating blood, this is what we're asking you to do before, when you have time today, we need to have a participant CRID and a matrix account in order to be able to donate blood. There are postcards on the tables, so if you have a minute before you come back to the back of the room, we're in the back of the this main floor on the New Jersey room. We're going to have you create a CRID and then fill out and make a matrix account where you can fill out your consent form in order to donate blood. That whole process takes about 20 minutes, so if you want to do that ahead of time, that'll save you a little bit of time once you get back to the room. So please scan the QR codes, create those accounts, and we're looking forward to helping you with your blood donations later today. That's all from me.
Thank you, Lindsay. Thank you so much.